We switch gears a little bit as we get into chapter 13, which is all about controlling microbial growth. Our last few chapters have been about how microbes grow and DNA replication and protein synthesis. So now we're going to look at what parts of each of those we can target and control the growth. In order to prevent the spread of disease, we have to control the growth of microbes on things that we touch, things that we use. And these are called fomites. So doorknobs, toilet handles, cell phones, keyboards, all of these things are called fomites because bacteria can, or other microbes can live on those, and they're able to transmit disease. The idea is that we need to control growth on all of these fomites so that we can cut down on the spread of disease from them. And there are two things that we look at for this. The first one is the application for which the item will be used. Okay, in other words, what are we going to use this fomite for? Is it a doorknob? Is it something that's, outside of the body? Or is it something that's going to go inside the body, like a catheter? Okay. And then, how resistant are those microbes to the materials or the methods that we're going to use to control growth? We know that a lot of microbes are resistant to different chemicals, and that has to be taken into consideration when we are trying to control growth. Now, when we look at people who work with pathogens, so, your mind automatically goes to the CDC. And, of course, in the United States, the CDC is, our foremost authority on microbes. And within these research facilities, there are what we refer to as biological safety levels, or BSL. And when we look at the levels of biological safety, we have to consider, how effective is that agent, how is it transmitted, and how severe is the disease. For BSL 1, this is the lowest level. It requires the least amount of precaution because these are the lowest risk microbes. When we talk about a BSL agent, we're talking about those that don't normally cause disease in a healthy human adult. Things like E. coli. Okay. In BSL 1, we practice aseptic technique. We can be out on an open bench or table. We don't need a hood. We can wear gloves. We can wear goggles. But really, there's not any other precautions that have to be taken. When we get up to BSL 2, then these pose a moderate risk. So these can be infectious. They can cause disease in normal, healthy adults. Things like Staph aureus, Salmonella, Shigella, different viruses like measles or mumps. BSL 2 laboratories have biological safety cabinets. They promote the use of an autoclave. There has to be an eye wash station, self-closing doors. Our lab is a BSL 2 because we do have autoclaves, we do have methods for disposal of the agents that we use, and we do work with pathogens. So we have a BSL 2, and most colleges are BSL 2s. Now, when we get up to BSL 3 and 4, we're talking about major infectious diseases. When we look at BSL 3, this is things that like HIV, West Nile virus, so they can cause lethal infections. Okay, there's restricted access. Only certain people are allowed in BSL 3s. They have to use a respirator. They work in a biological safety cabinet, so basically a fume hood, essentially. A hands-free sink. There has to be an eye wash station, and not only do they have to have self-closing doors, they have to have self-locking doors as well to restrict that access. They also have to have a fresh supply of air, so air is not recirculated in the BSL-3s. BSL-4 is our most dangerous types of infections. They're easily transmitted by inhalation. They cause diseases where there are no treatments. These are things like Ebola and Marburg, which are hemorrhagic fevers. We have very few BSL-4s in the United States. There's one at the U.S. AMRID, which is the Army branch for infectious diseases. There's one at the CDC, and there's one in Colorado at our vector-borne disease lab. So, and there may be a couple of others scattered around, but these are a very high security, very restricted access. The workers in a BSL-4 lab have to change their clothes when they enter. They take a shower when they exit. They have to wear these special suits, full body suit. They have HEPA filters, HEPA exhausts, and the facility is going to be on its own air system, its own air supply and exhaust system. And so this would be, someone working in a BSL-4, they're going to have these, pressurized suits with their own air supply. They're going to be hooked up to these hoses as they walk around the lab so that they can have their own air supply. They're not actually breathing in the room air. And they would shower upon exiting just to make sure they wash all of those microbes off of them. Now let's discuss some of the terminology that we're going to use as we consider methods to control growth. Sterilization is the removal of all vegetative cells, endospores, and viruses. So we are going to make something sterile, completely free of any and all microbes. And the chemicals that achieve sterilization are called sterilants. And sterilants will kill microbes and viruses, and with a long enough exposure time can also kill endospores. So that's why they're referred to as sterilants, because they do achieve sterilization. As you've seen in the lab, we practice aseptic technique. We don't want to contaminate our samples, and so we use the flame to achieve sterilization of the loop, and we also use the flame to sterilize around the mouths of the tubes. So we practice our aseptic techniques every time we're in the lab. We also have commercial sterilization, which is when we use temperatures. We use heat at the temperature that's low enough that it's not going to affect the quality of the food, but it's got to be high enough that it can destroy the bacteria that are responsible for food poisoning, such as Clostridium botulinum or Clostridium perfringens. So it's not really sterilization because we're not getting rid of all microbes. That's why milk still spoils if you leave it in your fridge long enough, even unopened. But it's getting rid of the pathogens that can make us sick. Most of the time, though, when we are trying to rid an area of microbes, we are not going to achieve sterilization. Most of what we do is disinfection, and this is an inactivation of most microbes. We're probably not going to get rid of the endospores, so there are going to be some microbes that remain. So it's not sterilization, it's disinfection, and we use disinfectants for those purposes. And we're going to look at a whole range of different disinfectants in a later section. The ideal disinfectant needs to be fast acting. We don't need to have to leave it on the surface very long. It has to be stable. We want it to be shelf stable and easy to prepare, so we don't have to go through these elaborate preparations. It needs to be cheap. We don't want to have to pay a lot for these disinfectants, and easy to use. Spray on and wipe off. Antiseptics are very similar in the manner in which they work to disinfectants, but antiseptics are safe to use on living skin or living tissue. Now, some antiseptics, like peroxide and alcohol, can also be disinfectants. They can be used on surfaces, but they're also safe enough to be used on the skin. The antiseptic also needs to be, cheap and easy to use, a quick exposure time, and able to get into the tissues without causing damage to the tissue. Degerming is when we scrub living tissue. So hand washing. When surgeons get ready to go into surgery, you see them at their scrub sink and they're scrubbing their hands, scrubbing their arms. That is degerming. We're using soap, so we're using some type of soap that helps to kill microbes, but also the process of degerming physically removes microbes from the surface. And then our other term we have here is sanitization. Sanitization is looking at public health levels. So this is cleaning fomites to remove enough microbes to get to a level that is safe for public health. So things like commercial dishwashers. Commercial dishwashers use much hotter water than we have at home, and they are very quick. When you put dishes in your dishwasher at home, it may take an hour or more. Commercial dishwashers can do the same thing in 5 or 10 minutes. Because they're using such hot water, it doesn't take as long. They also typically use hot air for drying, and so this is going to kill most microbes, and that is referred to as sanitization. You've got a table here in section 13.1, which is figure 13.4, that summarizes all of these different protocols for you.